Hello ASBS conference. Sorry I can't be there in person, but I am stuck here in sunny, warm Edinburgh, and I promise you there's nothing more than where I'd rather be than in Alice Springs at the conference right now. But I'm sorry that isn't possible at the moment. So what I'm going to give you here is I'm going to give you kind of an update and a run through of where we're at with the actual project itself at the moment, because we've made some great progress. However, we haven't got solid results at the moment just because the analysis has taken a little bit longer than expected. So let's begin. So cycads represent the world's most endangered organisms. More than 60% of cycads are recognised as extinct in the wild, extinct or endangered or critically endangered by the IUCN, which is quite significant, and we need to try and preserve these plants for the greater good. So Australia represents a biodiversity hotspot for cycads. We have 4 genera of cycads in Australia, both families, Cycadaceae and Zamiaceae. And Cycas is a pretty large genus represented with 38 species in Australia, which is quite important. Australia's populations are perfect to look for genetic studies because these populations are relatively less disturbed and are often larger than we find elsewhere around the world. That's of all cycads in general. So with regards to population or conservation genetics, why do we use it? Well, we can use it to identify species. So we look at species in their habitat, and we can see differentiation between 2 species or genetic cutoff, where a species is either speciating into a subspecies or potentially a species itself. We can also use it to differentiate populations. You can see from the figure on the right here, which each individual coloured box represents an individual genotype. And you can see here there's a complete cutoff between the north and south of the island with Cycas micronesica. We can also use genetic data to help conserve wild populations. And this is great because it can allow us to look at cultivated collections, and do these ex situ conservation collections represent the genetic diversity of the wild populations, and how can we support that? However, there's no population genetic studies done on cycads in Australia, which is peculiar, or at least non-published at least. So this is our study species. This is Cycas calcicola. This is a beautiful plant, really attractive. From a distance it looks quite glaucous. However, it's not glaucous, it's got very dark green leaves, but it's covered with this beautiful silvery hair. And characteristically, this species grows on limestone. This is the typical sort of megasporophyle structure of Cycas micronesica with the seeds. And this is Cycas micronesica growing on sandstone, which should grow on limestone. This is growing on sandstone. Still beautiful species, really nice habitat around the Litchfield National Park. So what do we aim to do in this study? So we want to look at the complete genetic diversity of Cycas calcicola in the wild. We want to see which populations hold the highest levels of genetic diversity and which are priority for conservation. We also help to use this data to assess ex situ conservation collections as well. So we will look at the genetic data of ex situ conservation collections and see if the wild populations represent the diversity in these ex situ conservation collections. If not, we will use the wild populations to improve the representation in the ex situ conservation collections. We also wish to assess the geographic and morphological geographic disjunction between Litchfield and between the Catherine populations. And we will also use a range of morphological characters to help further identify populations. So we're not just looking at genetics here, we're also looking at morphology. So the fieldwork itself was conducted in Northern Territory in Darwin, and between Darwin we captured population in Litchfield National Park, which you can see here, and in the Catherine region. And for Cycas calcicola we captured a total of 6 populations, 60 individuals. We were aiming for 100, but this species was far more scarce in the wild than we hoped it was. We also captured this along with Cycas armstrongi, Cycas maconicae, and a hybrid between Armstrongi and McConaughey. So here we have the same figures again, and you can see in better detail here these typical ones in the Litchfield National Park region and the Catherine region, and there's a big disjunction between them. It's about a 4-hour drive from what I remember. So what we're using RAD sequencing. And I know some of you are already familiar with RAD sequencing, so I'll just go through it really, really quickly. So we start with the actual extraction itself, and then we use a series of enzymes to digest the genomic DNA into smaller fragments, and we size select these fragments, and then we adylate the ends, we perform end repair, and then we use a library preparation in regards to a Illumina kit, so Illumina TrueSeq Nano kit in our case, which we adapt dual index barcodes to the ends of the fragments so we can later identify them. Then we carry out PCR to amplify these, and we pull them into a single reaction. So we're actually effectively multiplexing these samples and then sequencing 96 individuals on a single Illumina sequencing run. In this case, we use NextSeq 500, which gave us good results, but probably a little bit scruffier data than what a HiSeq would do. But for the amount of data you get for the price, it was good. So I actually had a lot of help here from Todd from Melbourne to help quality check in some of these reads, because I had a few problems with some of the software I was using where I was using a lot of data or wasn't getting enough clusters. So actually I needed to do some good quality filtering and things with this data to get to work. So I said we sequenced using the NextSeq 500, so we use 150 paired end read. So we're reading 150 base pair fragments from either end. So if a fragment is 200 base pairs long, we're reading 150 in the forward direction, 150 base pairs in the reverse direction, so we don't have an intermate distance in that case. But if we have fragments of 200 base pairs, we're going to get a significant intermate distance. But how we get over that is by using a bioanalyzer read to see what our average fragment distribution. And ours is about 530 base pairs, which is actually quite nice and safe. So from total of what we did in total, we totally sent 288 samples off. This is not just calcicola. And we had 284 successful samples from sequencing, which is pretty effective, really. From the actual reads themselves, we firstly used PrintSeq to quality check the reads and see where we have a fall off in quality, because we found that some of the first few bases were a bit lower quality and the end bases were lower quality as well. And then we used Trimmomatic to trim off these reads and perform a sliding window and remove reads of less than 50 base pairs in length, so we could continue this. And then once we've filtered all these reads, what we thought we would do is we would align this to a reference genome, actually a reference chloroplast and mitochondrial genome, to see where our reads actually lie in the genome. And you can see just here, this is the reference sequence of Cycas titarensis, and this is Cycas calcicola mapped to titarensis. So this is from the chloroplast. So from the chloroplast, you can see that we've got from 3 million reads, only basically 110,000 map to the chloroplast genome and only 76,000 the mitochondrial genome. That means we have over 3 million reads which are unmapped, and this is probably in the nuclear genome, and this is where we want. As an example, this is what we need really here in this case with Cycas, because the Cycas genome isn't really easily sequenceable at the moment, because the genome itself is around 25 to 30 gigabases in length, which is a pretty big genome. So with regards to de novo assembly, this is the next bit. This is actually how we worked with the data. We use a piece of software called iPyrad. And iPyrad is basically a newer version of Pyrad based on a new code with some new features added in and a little bit more easier to work with user interface. So we first of all get the reads, we filter them, it merges the paired end reads together. We cluster these reads into basic what we call high-depth clusters, so it clusters the individual reads which overlap. Also, it estimates heterozygosity and the error estimations as well from each individual sample. And what we got is an output overall between 2,000 to 11,000 informative linked SNP, SNPs, which is really good. So, but however, we do have some peculiar. So this is, first we go, you can see here, this is for 4 samples. This is the raw reads we got. These are the merged pairs we have themselves. And then these are how many clusters we get. But if you notice, our number of high depth clusters are far lower than you would hope them to be. This is purely down to the genome size. And there's no real way around this with Cycas. Theoretically speaking, you could do a deeper coverage depth, deeper sequencing. We was aiming for around a gigabase per sample, which is quite a good amount. However, does it give us bad results? No. We get anything from, say, around 2,000 to up to 11,000 markers. So here we can see 1,900, 6,900, 2,700, 5,700, which is still quite a lot of nice informative markers. And if you think about previous populations, many population studies use probably 20 or 30 markers if they're lucky. So here we have a lot of markers to look for to find informative data from. What do we hope to achieve? So we're going to identify the populations with the highest levels of genetic diversity and those which are the priority for conservation. We're also going to compare how these ex situ conservation collections represent the genetic diversity that you find in the wild and build a conservation management plans. And we're going to look for disjunction between the Litchfield and the Catherine populations. So are these ones at Catherine, are they all the same species, or do we have some significant differentiation between these? I'm going to use morphological characters to further identify these. Where we're at with this is we're basically looking at the software now to kind of determine this and figure out the software packages. We do have to rerun one of the analyses for iPyrad because we had a number of bugs that we found in the software that we actually discovered when we was doing it, which have been corrected now. So going forward with this, it should work out well. So morphology-wise, we gathered a load of population data, you know, in sex ratios, the length of leaves, the length of leaflets, the number of sporophylls, the number of stems, the length of the caudex. And we also got the characters, the megasporophyll characters, length, width, terminal lamina shape. And we're also gathering a load of leaf cuticle data as well, which I'm actually in the process of now, using a scanning electron microscope to image leaf cuticles already prepared of Cycas calcicola. So we're going to look not just genetically, but morphology as well, and bring the molecular data, the morphological data together to see further differentiation between these. I've got to thank the funding and support we received for this project, particularly the ASBS, and the Nature Conservationary and the Thomas Foundation. We very much appreciate that. And also the funding we received in the UK as well, and the support. Any more questions? I would like to say that please ask you some questions, but it's kind of difficult at the moment, but you're welcome to just send me some questions on Twitter and I'll try and answer them from Twitter. However, I will also put this video through my blog, and you can always ask me questions for the blog and things. I can try and reply to you. There might be some way of video linking this, or I should say, give me a call through FaceTime or Skype, and I don't know what time it's going to be in the morning here. So I really apologise for not making it to the conference itself, but I really do appreciate the help of the ASBS and the people that have helped me here in the garden in Sydney and helped me do the facilities, which I greatly appreciate. I also really appreciate Todd's time and effort in helping me quality check and understand what was going wrong with my data back then. So thank you so much, and I really hope you guys enjoy the rest of your conference. Goodbye.